Okay, starting chapter 5. Try that again. Okay, chapter 5 is all about the cell membrane and transport. The plasma membrane separates the living cell from its surroundings. It exhibits selective permeability, allowing some substance to cross it and others to not cross it. Phospholipids are the most abundant lipid in most membranes. Phospholipids are amphipathic molecules, which means that they have both hydrophobic and hydrophilic regions. A phospholipid bilayer can exist as a stable boundary between two aqueous compartments, so water. Okay, so you should be familiar with this picture. This is your phospholipid bilayer, which is attached to your extracellular matrix, which is outside the cell, and the cytoskeleton, which is inside the cell. Notice some of your molecules here. You have a carbohydrate attached to a protein, making up a glycoprotein. Here we have a glycolipid where the carbohydrate is attached directly to the phospholipid, both of which function in cell-to-cell recognition. Notice here you have cholesterol, which is embedded within the cell membrane that helps to maintain the fluidity of the cell membrane. You have peripheral proteins, which are just on one side of the protein, one side of the cell membrane. You have integral proteins, which go all the way through. And I think that's it for that slide. Most membrane proteins are also amphipathic and reside in the bilayer with their hydrophilic portions protruding. The fluid mosaic model states that the membrane is a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. Groups of certain proteins or certain lipids may associate in long-lasting specialized patches. Okay, so here's your phospholipid bilayer. It's amphipathic, which means that this part is hydrophobic, the tails, which consist primarily of carbon and hydrogen bonds. And then your hydrophilic heads, which point out where there's water, and in toward the cell, where there is also water. Your cell is primarily made up of water. Most of the lipids in some proteins in a membrane can shift about laterally, so they can move side to side. They just can't flip from one side to the other. The lateral movement of phospholipids is rapid. Proteins move more slowly. Some proteins move in a directed manner. Others seem to be anchored in place. We're going to skip that. As temperatures cool, membranes switch from a fluid state to a solid state. The temperature at which a membrane solidifies depends on the types of lipids. A membrane remains fluid to a lower temperature if it is rich in phospholipids with unsaturated hydrocarbon tails. Membranes must be fluid to work properly. They are usually about as fluid as salad oil. Okay, so here we have unsaturated fatty acids. Remember that unsaturated lipids or fatty acids have at least one double bond, causing a bend in the molecule. So as a result, this is going to, in a cold environment, prevent the membrane from packing tightly together. Okay, because if it gets too cold, then it's going to become too viscous and it's not going to be able to work properly. So as your temperature decreases, you want to have more unsaturated fatty acids to prevent tight packing. As your temperature increases, the increase in temperature is going to cause your membrane to become more fluid. So as a result, you would want to have saturated tails as a result. Cholesterol reduces membrane fluidity at moderate temperatures, but at low temperatures it hinders solidification. So cholesterol also plays a role in the fluidity of the membrane. The steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperatures, cholesterol restrains the movement of phospholipids, and at cool temperatures, it maintains fluidity by preventing tight packing. Variations in lipid composition of cell membranes of many species appear to be adaptations to specific environmental conditions. The ability to change the lipid compositions in response to temperature changes has evolved in organisms that live where temperatures vary. So based on the temperature, the environmental temperature, they can switch from having unsaturated fatty acids in their membrane to saturated fatty acids and vice versa. A membrane is a collage of different proteins, often grouped together, embedded in the fluid matrix of the lipid bilayer. Proteins determine most of the membrane's specific functions. Integral proteins penetrate the hydrophobic interior of the lipid bilayer. Integral proteins that span the membrane are called transmembrane proteins. They go all the way through. The hydrophobic regions of an integral protein consist of one or more stretches of non-polar amino acids, often coiled into alpha helices. Peripheral proteins are loosely bound to the surface of the membrane. Okay, so here's a transmembrane protein. So the hydrophobic portion of this protein is here, because this is the hydrophobic portion of the membrane, and the hydrophilic regions are here and here. There are 6 major functions of membrane proteins. They include transport, enzymatic activity, attachment to the cytoskeleton or extracellular matrix, cell-to-cell recognition, intercellular joining, and signal transduction. Okay, so here we have transport proteins embedded within the membrane. They transport molecules into and out of the cell. You have enzymatic proteins, which catalyze some sort of chemical reaction. You have proteins that attach to the cytoskeleton or the extracellular matrix. You have glycoproteins that function in cell-to-cell recognition, proteins that join two cells together, and here we have a receptor protein, which binds to a signaling molecule, which leads to a signal transduction pathway, which leads to some sort of cellular response. Cells recognize each other by binding to surface molecules, often containing carbohydrates, on the extracellular surface of the plasma membrane. Membrane carbohydrates may be covalently bonded to lipids, forming glycolipids, or more commonly to proteins, forming glycoproteins. Carbohydrates on the external side of the plasma membrane vary among species, individuals, and even cell types in an individual. Membranes have distinct inside and outside faces. The asymmetrical distribution of proteins, lipids, and associated carbohydrates in the plasma membrane is determined when the membrane is built by the ER and the Golgi. So again, this shows synthesis of membrane components and their orientation in the membrane. A cell must regulate transport of substances across cellular boundaries. Plasma membranes are selectively permeable, regulating the cell's molecular traffic, only allowing certain molecules to get through, to get in and out. Hydrophobic molecules or nonpolar molecules, such as hydrocarbons, can dissolve in the lipid bilayer of the membrane and cross it easily. So they can just diffuse through pores in the cell membrane because they can make it through the hydrophobic core of the membrane. Polar molecules, such as sugars, don't cross the membrane easily because they can't get through that hydrophobic core. They can't get through the hydrophobic tails of the membrane. Transport proteins allow passage of hydrophilic substances across the membrane. Some transport proteins, called channel proteins, have a hydrophilic channel that certain molecules or ions can use as a tunnel. Channel proteins, called aquaporins, facilitate the passage of water. So remember that water is small enough to diffuse through the pores in the cell membrane, but in order to facilitate the transport of a large amount of water, the cell can use these aquaporins. Other transport proteins, called carrier proteins, bind to molecules and change shape to shuttle them across the membrane. A transport protein is specific for the substance it moves. Diffusion is the tendency for molecules to spread out evenly into the available space. Although each molecule moves randomly, diffusion of a population of molecules may be directional. At dynamic equilibrium, as many molecules cross the membrane in one direction as in the other. Okay, looking at the top here, we're starting with molecules at a high concentration of one side of a semi-permeable membrane. And as time progresses, those molecules are bouncing around. Some will go through the pores in the membrane, some will hit the membrane and bounce back. But if enough time transpires, you'll have an equal amount of solutes on each side of the membrane. That's dynamic equilibrium. The molecules are still moving, but you should have an equal amount of molecules on both sides of the membrane. Now, part B looks at two different molecules. Each molecule will move down its own specific concentration gradient. So the yellows, overall net diffusion will be to the right. The purples, overall net diffusion will be to the left. You have to look at the molecules individually, not as a whole. Substances diffuse down their concentration gradient from where they are more highly concentrated to where they are less concentrated. No work must be done to move substances down their concentration gradient. The diffusion of a substance across a biological membrane is passive transport because no energy is used to make that happen. Osmosis is the diffusion of free water across a selectively permeable membrane. Water diffuses across a membrane from the region of lower solute concentration to the region of higher solute concentration until the solute concentration is equal on both sides. Okay, so here we have a semi-permeable membrane. We have more solute on this side. We zoom in on that at the molecular level. So these red and white things, these are the water molecules, and the green is the sugar. Now notice that because you have more sugar on the right, you have less free water. Free water is water that is not doing the dissolving. So we say that water moves from where there is more free water to where there is less free water. So water moves down its concentration gradient. It moves from where it is more highly concentrated to where it is less concentrated. Now the confusion is, you could also say that water moves to where there is more solute. So you have to be specific when you're talking about what you're referring to. Are you referring to the sugar or solute molecules, or are you referring to the water molecules? So water will move from where water is highly concentrated, more free water, to where there is less free water. Or water is going to move to where there is more solute. Tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. That's my son ringing the doorbell. Isotonic solution, solute concentration is the same as inside the cell. There's no net water movement across the plasma membrane. Hypertonic solution, solute concentration is greater than that inside the cell loses water. And hypotonic solution, solute concentration is less than that inside the cell, and the cell will gain water. Okay, well, what does that look like? Here we have an animal cell and a plant cell, both in a hypotonic solution. So there's going to be more solute on the inside of the plant and animal cells than the outside. Excuse me for a minute. I'm not done. What?
This is what and I want to come in here.
Okay, you have to be quiet, though, until Mommy's done. Can you do that? Okay, come on in.
Daddy wants it.
I know. Okay. So for an animal cell in a hypotonic solution, close that door too. Water will enter the cell and the cell will lyse or burst. Now, plant cells are different because they have a cell wall. So a plant cell that is in a hypotonic solution will become turgid because the water will enter the cell and the cell wall will prevent the cell from bursting. In an isotonic solution, this is the ideal environment for an animal cell because water is entering and leaving at the same rate. For a plant cell, it is not ideal because the plant cell needs to have a lot of water in order to undergo photosynthesis. If it has the same amount of water entering as is leaving, the cell will become flaccid. Now, when placed in a hypertonic solution where there is more solute on the outside of the cells than the inside, this is bad for both animal and plant cells. In an animal cell, water will leave at a higher rate and the cell will shrivel and die. A plant cell, same thing, water will leave and plasmolysis will occur where the cell membrane pulls away from the cell wall. Hypertonic and hypotonic environments create osmotic problems for organisms. Osmoregulation is the control of solute concentrations and water balance, and it is a necessary adaptation for life in such environments. The protist Paramecium caudatum, which is hypertonic to its pond water environment, has a contractile vacuole that can pump excess water out of the cell. So we talked about this last chapter, where this is a paramecium, a protist, that's in a hypotonic environment, so lots of water, not a lot of solute. It has more solute inside of its cell, so water rushes into the paramecium, and the paramecium uses its contractile vacuole to pump water out. And then this is all sort of a repeat of everything that I just said in the prior slide, so I won't repeat that. Facilitated diffusion is a type of passive transport that's aided by proteins. In facilitated diffusion, transport proteins speed the passive movement of molecules across the plasma membrane. Channel proteins provide corridors that allow a specific molecule or ion to cross the membrane. Channel proteins include aquaporins, which facilitate the diffusion of water, and ion channels that open or close in response to a stimulus, gated channels. You saw these when we just talked about the nervous system. So here's a channel protein. Now, even though proteins are involved, it's still passive transport, still high to low concentration. Carrier proteins change shape. These are found, for example, within your nervous system, your sodium-potassium pump. Carrier proteins undergo a subtle change in shape that translocates the solute binding site across the membrane. The shape change may be triggered by binding and release of the transported molecule. And again, because this is passive transport, no energy is required for facilitated diffusion, right, Lucy?
Yeah.
Yeah. Okay. All right. So I guess since my children have found me, we're going to conclude this part of Chapter 5.
Mommy?
Yes, buddy.
So